multiple alleles and the blood groups. ABO blood system is the blood system of the humans. ABO blood system is controlled by a gene. That gene is triallelic in nature. This gene contains 3 alleles, allele A, allele B and allele I. This gene can generate 6 different genotypes. Out of the 3 alleles, 2 alleles they are codominant to each other. Allele A and allele B, when they exist together, they are codominant to each other. They do not dominate over the other. Allele A cannot dominate over the allele B and allele B cannot dominate over the allele A. Such a phenomena is called as codominance. While these both allele A and allele B, they are dominant over the third allele. Allele A and allele B, when they exist together, it is called as codominance. These alleles, which are allele A, allele B and allele I, they control the antigens, or they are responsible for the production of the antigens which are present on the surface of the red blood cells. Our blood have 3 types of the cells: red blood cells, white blood cells and platelets. And these alleles, they generate antigens. Those antigens, they are present on the surface of the red blood cells. So here we can see that alleles and the antigens. Allele A, that generates an antigen that is called as antigen A. Here we can see the antigen A. Antigen A is present in the membranes of the cell membrane of the red blood cells. So this is the membrane, cell membrane of the red blood cells, and within the red blood cells there are these antigens. These antigens, they are called as antigen A. While in case of allele B, it produces antigen B, and antigen B is also present in the cell membrane of the red blood cells. These are the antigen B. While in case of the third allele, that is allele I, that allele I produces no antigen. When there is allele I, there will be no antigen on the surface of the red blood cells. Here we can see the cell membrane of the red blood cells, and there is no antigen present on the surface of the red blood cells. So in this diagram we can see how these antigens, they are different from each other. In case of first blood group, that is blood group A, in case of blood group A, the antigens, if we mark them with the help of triangles, they are in the form of, let's say, they are in the form of the triangles. In case of allele B, on the surface of the red blood cells, there are the antigens. Those antigens are called as antigen B, and antigen B, they can be marked as circles. So these are the antigen B. While in case of AB blood group, there are both the antigens which are produced by allele A and antigen which are produced by the allele B. In this diagram, we can see that on the surface of the red blood cells, both type of antigens are present, antigen A as well as antigen B. So this phenomena is called as codominance, because in case of AB, both the antigens are present on the surface of the red blood cells, while in case of blood group O, the red blood cells, they contain no antigen. So in case there is no antigen on the surface of the red blood cells, no antigens are present on the surface of the red blood cells. So based on the genotypes and phenotypes, if we see that based on the alleles, there are 6 different genotypes, and based on the genotypes, there are 4 major groups of the human blood. In case of genotype, if there is a genotype A and A, mean both the alleles are A, and that is in the form of homozygous, then the blood group will be A. while in case of it can be in the form of homozygous and it can be in the form of heterozygous. So this is the example of heterozygous. Here one is allele A while the other is allele O. So this is in heterozygous form, but the phenotype will be same, that is A. So A blood group have two genotypes, one in homozygous form, that is homozygous, and one in heterozygous form, this is heterozygous. Same is with the blood group B. Blood group B is in two forms, that is in homozygous form and in heterozygous form. Here we can see that both alleles they are B and the blood group is B, and this is in homozygous form, B in homo, and here B is in hetero form. And on the other hand, AB blood group just has one genotype, single genotype, and O blood group also have single genotype. So based on the comparison between genotypes and phenotypes, we can conclude that there are 6 different genotypes and there are 4 different blood groups of the humans. Although blood group they exist in positive and negative form. Blood group A can be positive, can be negative. Blood group B can be positive, can be negative. But it depends on the Rh factor which is present on the cell membranes. So blood group exist in, there are three blood groups: blood group A, blood group B, blood group O, and blood group AB. Blood group A exists in two forms, that can be in homozygous form, that can be in heterozygous form. Blood group B also exists in two forms, that can be homozygous, that can be heterozygous. But the blood group AB and the blood group O, they have only single genotype. So we can conclude that the blood group system in the humans is controlled by a gene. That gene has 3 alleles: allele A, allele B, and allele O, or allele I small I. It is represented by O or written by as I. So there are 3 different alleles. They generate 6 different genotypes and 4 different phenotypes. But based on the Rh factor, the 4 different phenotypes, they can be in positive form and in negative form. Like the example is A blood group can be positive and A blood group can be negative. But this positive and negative is based on the Rh factor.